There are two types of signaling processes. First is electrical signaling that passes through gap junctions that allow local current to flow across in either or both directions from an arriving action potential. This type of signaling is extremely rapid compared to the second one, which is chemical signaling. Chemical signaling is relatively slower because the synaptic delay is due to the time necessary for the transmitter to be released from presynaptic membrane along the cleft to postsynaptic membrane. There are three different types of chemical signaling, namely endocrine signaling process, paracrine signaling process, and autocrine signaling process. The endocrine signaling process involves substances called hormones. These are released in the bloodstream by the organ or tissue that produced them, and these hormones will then be carried to their distant target organs for them to produce a specific response. The paracrine signaling process involves cells that release their neurotransmitters to adjacent cells through diffusion. Cells in the autocrine signaling process release their neurotransmitters to act on themselves. Synaptic transmission occurs once action potential reaches the axon of the neuron. As the signal reaches the axon terminal, the voltage-gated calcium channels open up. This facilitates the entrance of calcium to the terminal. Then, the vesicles containing the neurotransmitter proceeds to the active site and eventually fuses with the plasma membrane. This releases its contents to the synaptic cleft. The neurotransmitter travels to the postsynaptic receptors of the postsynaptic cell. There are two types of receptors which are found in the postsynaptic cell. The first type is ionotropic receptors or ligand-gated ion channels. These are rapid protein channels that open or close in response to the binding of a chemical signal. Therefore, they allow the passage or non-passage of an ion via a single receptor and effector. The second type of receptor is the metabotropic receptor. These receptors are indirectly gated since the receptor and the effector of the channel are different. Therefore, they are slower compared to ionotropic receptors. There are two types of metabotropic receptors, namely G-protein linked receptors and tyrosine kinase receptors. G-protein linked receptors are those that function through a single polypeptide chain with 7 membrane-spanning alpha helical segments. The G-protein with an intrinsic GTPase activity is composed of 3 subunits, namely alpha, beta, and gamma subunits, of which beta-gamma subunits serve as the regulatory component. There are 3 different pathways in this type of receptor. The first type is the cyclic AMP pathway, wherein the first messenger norepinephrine binds to the beta-adrenergic receptor of the G protein. This will make GDP to be displaced by GTP on the alpha subunit. This displacement will in turn make the G protein dissociate into alpha subunit with GTP and the regulatory beta-gamma subunits. The activated protein G-alpha then stimulates the primary effector adenyl cyclase to convert ATP to cyclic AMP or CAMP, which acts as a secondary messenger. CAMP will then interact and cause changes in the activity of the system's secondary effector or protein kinase A. However, there are 2 G protein types. One is the GS type, which is stimulatory. that produces result, while the other is the GI type, which is inhibitory to produce no effect. The fate of the beta-gamma subunits will be either to activate ion channels such as potassium channels or to activate other effectors. The termination of response is via the autohydrolysis of GTP to GDP, and therefore it will bring back the reassociation of the alpha, beta, and gamma subunits. In patients suffering from wheezes, salbutamol is given. This reacts with the beta-adrenergic receptor of the G protein that results in the activation of the CAMP pathway. As a result, bronchodilation of the pathways occur. The second pathway of G protein linked receptor is the phosphoinositol pathway. This pathway is activated by the hydrolysis of inositol phospholipids of the plasma membrane. Agonists such as acetylcholine or epinephrine bind to muscarinic ACH receptor of the G protein to activate GQ. The activated GQ stimulates the activity of phospholipase C-beta to hydrolyze phosphatidylinositol 4,5-biphosphate. The cleavage of the phosphatidylinositol 4,5-biphosphate results in the release of IP3 and diacylglycerol, DAG, both of which are secondary messengers. IP3 reacts to a specific calcium receptor channels in the endoplasmic reticulum, causing calcium to be released. The released calcium will then bind to the cytosolic protein kinase C, causing it to bind with the cytoplasmic surface of the plasma membrane. Calcium, together with DAG, activates protein kinase C to phosphorylate important cellular effector proteins. In cases of hypotension, epinephrine is given to the patient to increase his or her blood pressure level. Epinephrine acts by binding to the alpha-1 receptor that activates the phosphoinositol pathway. The calcium released from the pathway through the IP3 second messenger reacts with calmodulin, resulting to muscle contraction and an increase in blood pressure. The final pathway of the G protein linked receptors is the arachidonic acid pathway. The first messenger, histamine, binds to the histamine receptor that will activate the unknown G protein. The activated unknown G protein stimulates the activity of phospholipase A2, which serves as the pathway's primary effector. Phospholipase A2 then acts on membrane phospholipids by cleaving the fatty acid on the carbon 2 of the glycerol backbone of the triacylglycerol. This phospholipase A2 mediated cleavage releases significant amounts of arachidonic acid, which is a secondary messenger. The released arachidonic acid serves as a precursor for the cellular synthesis of the secondary effectors 5-lipoxygenase, 12-lipoxygenase, and cyclooxygenase. The drug aspirin works through the inhibition of cyclooxygenase of the arachidonic acid pathway. This reaction results to the drug's anti-inflammatory, analgesic, antipyreptic, and antiplatelet effect. The second type of metabotropic receptor is the tyrosine kinase receptors. These receptors exist as a single membrane spanning polypeptide. If a signal molecule binds to the binding site, two polypeptides aggregate and form a dimer. Using phosphate groups from ATP, the tyrosine kinase region of each polypeptide phosphorylates the tyrosines on the other polypeptide. Once it has been fully activated, the receptor protein binds the specific intracellular protein. This then attaches to a specific phosphorylated tyrosine, resulting to an initiation of a signal transduction pathway. Since cellular division is activated when ligand binds and activates this kind of receptor, their inappropriate activation may result to uncontrolled cell growth or cancer.